జయదేవ్. హలో జయదేవ్. హలో జయదేవ్. హలో
జయదేవ్ షేర్ అవుతుంది చూడు ఒకసారి. వాయిస్ వినిపించ లేదు 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 ఆన్ చేశాను నేను. చూడు ఒకసారి చూడు, నేను ఆఫ్ చేస్తాను. ఇదా చూస్తాను. ఇప్పుడు షేర్ అవుతుంది చూడు ఒకసారి. షేర్ అవుతుంది చూడు ఒకసారి. వాయిస్
ఇప్పుడు అన్మ్యూట్ అయిందా?
హలో వాయిస్ వినిపిస్తుంది, అది ఇందులో రావట్లేదా? ఆయన ఇది ఒకసారి అయితే కనెక్టింగ్ డివైస్ ఒకసారి ఆన్ చేయి.
అన్మ్యూట్ అయిందా?
మీ దాంట్లో ఒకసారి డివైస్ టు కనెక్ట్ అని ఉంటుంది చూసారు, ఫస్ట్ ఆడియో అది ఒకసారి అది ఆన్ చేయి. నీది వస్తుంది, ఇందులో రావట్లే. ఒకసారి చూడు, ఇందులో దీంట్లో ఒకసారి మాట్లాడు. ఇది ఫోన్ కట్ చేస్తాను. ఇటు పక్కన చివరిన కనెక్టింగ్ త్రూ అని ఉంటుంది ఫస్ట్. కనెక్టింగ్ డివైస్ అది డివైస్ టు కనెక్ట్ అని ఉంటుంది చూసారు, ఫస్ట్ ఆడియో
హలో
వాయిస్ క్లియర్ ఉంది సార్.
నాది వాయిస్ పైన వస్తుంది, నీది ఒకసారి కనెక్టింగ్ అని ఉంటుంది చూడు దాంట్లో. జయదేవ్
చివరిన కనెక్టింగ్ త్రూ అని ఉంటుంది ఫస్ట్.
హలో హలో జయదేవ్, నీది ఒకసారి కనెక్టింగ్ అని ఉంటుంది చూడు దాంట్లో. జయదేవ్
అవుతుందా? దాంట్లో వాయిస్ రావట్లే. రావట్లే. ఏం చేస్తారంటే ఇప్పుడు ఈ చివరిన రైట్ అదే లాస్ట్ లోనే మీరు కనెక్టింగ్ డివైస్ అని వచ్చిందా? జస్ట్ మైక్ అని వచ్చింది నీ దాంట్లో. ఆడియో ఉంటుంది చూసారే, దాన్ని ఒకసారి క్లిక్ చేయి. దాంట్లో కనెక్ట్ అవుతుంది. జస్ట్ హెడ్ ఫోన్స్ కానీ అయితే చివరిన అది క్లిక్ చేయి. మైక్రోఫోన్ కానీ లేకపోతే డివైస్ కానీ. అయిందే. అక్కడ గ్రీన్ కలర్ ఉంటుంది మీకు, యారో మార్క్ వచ్చి ఇక్కడ. నీకు మైక్రోఫోన్ ఉందా నీ దాంట్లో? పెట్టావా? అవే వద్దు, డైరెక్ట్ గా జస్ట్ మీరు అది ఒకటే కింద బార్ లో ఉంటుంది, కింద ఉంటుంది చూసారా టూల్ బార్, దాంట్లో ఈ లాస్ట్ ఏదైతే ఉందో చివరిన మైక్ వచ్చిందని దాంట్లో మైక్ సింబల్. ఆడియో నొక్కు ఒకసారి, పైన కనెక్టింగ్ డివైస్ త్రూ ఆడియో అని వస్తుంది, అది అయితేనే మనకి దీనికి కనెక్ట్ అవుతుంది, లేకపోతే అవ్వదు. ఒక్కొక్కసారి డైరెక్ట్గా అవ్వాలి. మరి రావట్లేదా? పెట్టు ఒకసారి. ఎక్స్ట్రా మైక్రోఫోన్ బయటది పెట్టు ఒకసారి. వినపడట్లే. ఒకసారి చెప్తాను సాయిరామ్ గారు. ఇది రావట్లేదు అంటున్నారు, వాయిస్ రావట్లేదు అంటున్నారు. ఒకసారి చూడండి.
అది మ్యూట్ అయి ఉంటుంది సార్ అక్కడ మ్యూట్ అనే ఆప్షన్
ఏదో అన్మ్యూట్ చేసుకో ఒకసారి నువ్వు. జయదేవ్ అన్మ్యూట్ చేసావా? వాయిస్ నువ్వు మాట్లాడేటప్పుడు అందులో గ్రీన్ వస్తుందా? నువ్వు మాట్లాడేటప్పుడు నీ స్పీకర్లో, నీ డిస్ప్లేలో నువ్వు మాట్లాడేటప్పుడు అది ఏం చేస్తారంటే మైక్రోఫోన్తో కనెక్ట్ అయ్యిందా? ఇయర్ ఫోన్స్ అని పెట్టుకున్నావు? ఇయర్ ఫోన్స్ అది పెట్టు ఒకసారి. నువ్వు ఏది నువ్వు ఆల్రెడీ ఉందా? ఇయర్ ఫోన్స్ ఉన్నాయా? ఒకసారి పెట్టి చూడు, దాంతో చూడు ఒకసారి. అది ఏమన్నా అది ఆన్ అయిందా? అది అవుతుందా అవ్వట్లేదు ఒకసారి చూడు. రావట్లే, నీ వాయిస్ రావట్లే. ఫోన్లో వినిపిస్తుంది తప్ప అందులో రావట్లే. ఒకసారి లాగౌట్ అయ్యి మళ్ళీ జాయిన్ అయ్యి ఒకసారి.
ఇది బెస్ట్ ఆప్షన్.
పోస్ట్ ఒకసారి మళ్ళీ ఇలా ఇచ్చేసాయి. ఇది
అక్కడ S.V.K.P మీద క్లిక్ చేసి
S.V.K.P మీద క్లిక్ చేసి ఒకసారి హోస్ట్ అని ఇచ్చాను.
మోర్ అనే ఆప్షన్ ఉంటుంది సార్. క్లిక్ చేసి
మోర్ ఆప్షన్ దగ్గర
మేక్ మీ హోస్ట్ అని రైట్ క్లిక్.
మేక్ హోస్ట్ అని ఇచ్చేసి నువ్వు ఒకసారి లాగౌట్ అయ్యి మళ్ళీ లాగిన్.
రైట్
S.V.K.P అని చూసారా?
రైట్ సార్. మనకు ఇప్పుడు హోస్ట్ సార్ మనం.
అయ్యారా?
హలో,
వస్తుంది వాయిస్ వస్తుంది.
ఇప్పుడు వస్తుంది వాయిస్?
వస్తుంది.
క్లియర్ సార్, క్లియర్.
ఓకే. మా అమ్మాయి వాడి ఆ అమ్మాయి పేరు మీద ఉంది. అది నేమ్ రీనేమ్ చేస్తున్నా. మా అమ్మాయి వాడదు జూమ్ కాల్స్ అయ్యి స్కూల్కి వాటికి, అందుకని ఆవిడ పేరు మీద పెట్టుకుంది. మళ్ళీ నేను ఇప్పుడు మార్చుకోవాల్సి వస్తుంది ఎక్కడికి వెళ్ళిన మీటింగ్కి.
వీడియో రావట్లే? వీడియో ఆన్ చేయండి ఒకసారి.
వీడియో చేస్తాను.
రైట్,
బాగానే వస్తుంది.
సో టోటల్ ఎంతమంది రిజిస్ట్రేషన్స్, ఎంతమంది ఉన్నారు ఏంటి? మనకి
150 అయ్యారు. అందులో 100కి మనకి ఉంది. తర్వాత మిగిలిన వాళ్ళు యూట్యూబ్ ఉంది.
ఓకే.
ఎంతమంది అవుతారు? 100 వరకు ఉంది. ఇంకేం చేయదు.
చాలా రోజులు అయిపోయింది చూస్తే సార్. ఫస్ట్ వస్తున్నా హైదరాబాద్.
సరే సరే, ఈ టైం వచ్చేటట్టు ఉందా?
చూస్తా, వీలైతే అటు వస్తా.
సరే సరే, లేకపోతే నేను వచ్చేస్తానట్టు.
రా హైదరాబాద్ రా. అదే నీదేదో వర్క్ ఏదో ఉంది, ప్లాన్ చేయాలన్నా.
అందుకని వస్తాను నేను అటు వస్తాను. ఇలాగ నాకు కూడా కొంచెం పని ఉంది.
ఓకే,
వస్తాను. అయితే ఎన్ని రోజులు ఉంటావు?
నేను 15th దాకా ఉంటాను.
ఓకే, వస్తాను.
16th మళ్ళీ రిటర్న్ ఫ్లైట్ నాది.
అయితే వస్తాను నేను. అయితే రికార్డింగ్ పెట్టలేదు కదండి ఇది.
ఇంకేంటి, ఎవరెవరు ఉన్నారు అసలు కాలేజీలో? మనకు తెలిసిన వాళ్ళు ఎవరెవరు మిగిలి ఉన్నారు?
ఇదండి
మన కాలేజీలో ఇంకా మనకు తెలిసిన వాళ్ళు ఇంకెవరెవరు మిగిలి ఉన్నారు అంటున్నా.
చూడు సార్
రోజు-
మన ఉండగా కృపారం గారు మన ఇంగ్లీష్ లెక్చర్ చూసే ఉంటా గుడ్ ఆయన చూడు.
సార్నే 23 ఏళ్ళు అయిపోయింది.
Yeah, this is Kripavaram, Department of English. Do you remember me?
I think so, sir, but not confident enough. Like it's already 23 years.
Yeah, I came in the year 1996. By the time you are in second year, I think.
Second year. We are in second year.
After Sash Kumar, I am the member.
Okay.
Perhaps.
శశి కుమార్ గారిని అయితే గుర్తుపడతారేమో మీరు గుర్తుపడతారేమో.
Okay, I congratulate you for being your good position. I came to know that you are in a good position.
Thank you, sir. So at
Thank
least
God,
most of our department, our batchmates are in very good positions. Most of us are in good positions,
Yeah. I came to know through Ramesh. You are the you are good batch. The students of good batch in those days. So wish you all the best.
Thank you, sir. I wish I could come there physically and do this.
Okay, most welcome. Most welcome.
Yeah. I was planning like almost 23 years or 24 years. I want to visit the campus and see how it changed.
A drastic change. There came a drastic change in the campus. We have gone for 3 times for NAC recreation, and the atmosphere, almost the environment is totally changed comparing to your days.
Yeah, hopefully it should be a progressive change.
We heartily welcome you to our college.
Sure, sir. Sure, sir.
Thank you. Thank you.
అందులో MBBT అని పడింది. చెప్పు.
One minute, Ramesh. Comments सबने लिख दिए ना? सबने लिख दिए। मैंने आपको बोल देना मुझे सिस्टम पे मेरे सिस्टम पे एक्सेस नहीं है ऑनलाइन साइन करने के लिए, तो मैं उस वाले सिस्टम पे बैठ के तो प्रिंट लेके आएगा तो मैं प्रिंट साइन कर दूंगा, तो मे बी आफ्टर 1 o'clock, ठीक है 1 o'clock तक उसके बाद वो प्रिंट लेके आ जाएगा तो साइन कर दूंगा, नहीं तो मैं आफ्टर 1 o'clock मैं सिस्टम का सिस्टम पे बैठ
రీసెర్చ్ నుంచి
हम तैयारी से दूसरी बच्चे और भैया सर प्रिंसिपल చెప్పాలి కదా రూల్ కట్టుకొని అందరినీ అవ్వాలి మా వాళ్ళు ఎవరో ఉంటున్నారు ఓకేనా ఎగ్జిక్యూటివ్
చరణ్ చరణ్
ఏమా ఎక్కడ ఆ ఫోన్ ఇచ్చాడండి
దా గిన్నెస్ పెడతారా ఆయమా
దా.
Well, good morning, sir.
Hello, Jaydev.
Yeah. Hello.
పార్టిసిపెంట్స్ వస్తుంది తర్వాత స్టార్ట్ చేద్దాం ఒకసారి అందుకు ఆగాను.
Yeah, sure.
Okay, sir.
పర్లేదు సార్, మొదలు పెట్టేద్దాం మళ్ళీ. అన్నారండి, చెప్పనన్నారండి. అదే కనెక్టివిటీ లేదు. ఓకే.
Good morning, all of you. Respected management of our college association, President Garu, Sri Petana Surnarayan Garu, Secretary and Correspondent Dr.K. Ramchandra Raj Garu, Treasurer Sri Uddagiri Lavkumar garu, and other governing body members, and speaker of today's webinar and chief guest of this program, Dr. Sri Krishna Jaidev Magani garu, Associate Professor, Department of Life Sciences, Shivanadar University, Noida. Head of the Department of English, IQAC Coordinator, S.V.K.P garu. Convener of this program, Head of the Department of Biotechnology, T. Ramesh garu. The faculty members of our college and students of our college, and the participants from various places of Andhra Pradesh. Very good morning to you all. As we all know that biotechnology is the integration of life sciences and engineering sciences. So it's the technology that utilizes biological systems or living organisms are part of these living organisms to produce or create a different product. So the process of brewing in breweries and the baking of bread are the simple examples of these biotechnological processes. So the biotechnology find numerous applications in various fields, including therapeutics, diagnostics, agriculture, food processing, waste management, energy production, and so on. So according to these applications, the biotechnology has been developed into sub branches that includes applied biotechnology, plant biotechnology, medical biotechnology, chemical biotechnology, and industrial biotechnology, so on. So as far as our topic is concerned, biotechnology and human health. So even though it has
Ramani garu, please unmute yourself. Ramani garu,
Mute.
mute yourself. Sorry, mute yourself.
even though the biotechnology has numerous applications, we are today concerned only the human health biotechnology and its application in human health. So regarding this one, we can use the biotechnology in human health in 3 ways. So with this biotechnological techniques, we can diagnose the disease, we can prevent the disease, and we can treat the disease. So with the simple test of ELISA, enzyme linked test, we can diagnose the disease with biotechnology. And by using vaccines, we can prevent the disease by giving the vaccines to the whole organism, partial organism, and recombinant. And we can treat the diseases using biotechnological techniques. Those are gene therapy, stem cells, and cloning. So with these few words of the applications of biotechnology, in concerned with the human health, I am closing, and I inviting the chief guest of today's webinar, Dr. Sri Krishna Jaidev Garu. Before he speaks, I invite HOD of Biotechnology and convener of this webinar, T. Ramesh Garu, to introduce our guest speaker. Thank you.
Thank you, sir. Good morning, ladies and gentlemen. On behalf of S.V.K.P and Dr.K.S Arts and Science College, Penugonda, management, principal, staff, and students, I extend a warm welcome to the webinar on biotechnology and human health, jointly organized by Department of Biotechnology, Zoology in association with IQAC. In human healthcare, biotechnology products include quicker and more accurate diagnostic tests, therapies with fewer side effects. So we know the biotechnology is one of the emerging technology. We can detect more diseases and medical conditions more quickly with a great accuracy, because new biotechnology, by using the tools of biotechnological diagnostic based tools. So in this connection, I would like to introduce today's guest speaker, a good friend of mine, Dr. Sri Krishna Jaidev Magani, to the participants. Dr. Jaidev presently working as an associate professor in the Department of Life Science, Sivanadar University, Greater Noida. He did his PhD work at Netaji's Institute of Technology, University of New Delhi. He did his postdoctoral research at National Institute of Immunology, NII, New Delhi. He did his graduation, post-graduation MSc at Guru Ghansdas University, Bilaspur. He did his BSc at S.V.K.P and Dr.K.S Arts and Science College, Penugonda. He published more than 15 various national and international journals. He published chapters in 6 books. He attended various conferences and workshops. 4 students were awarded PhD under his guidance. He is not only active in the academic activities, he is a good sportsman, and he's the lead. He lead volleyball team for AIMS for 6 years, from 2002 to 2007. Won 3 gold medals and 2 silver medals. With these few good words, I present Sri Krishna Jaidev to the August gathering. Now I request Principal of our college, Chairman of today's function, Dr. Y.V. Apparao Garu, to continue the proceedings. Thank you.
Thank you, Ramesh Garu, for your nice introduction of our guest speaker, Dr. Jaidev Garu. Thank you. Now I invite Dr. Jaidev Garu to continue his lecture. Thank you.
Dr. Ramesh, can you just transfer the whole slides to me so that I can present mine?
Doing, sir.
Yeah, sure. So I thank you all for inviting me here. So I thank the management and the principal of the S.V.K.P and Dr.K.S Arts and Science College. So it's a pleasure to present in front of my own college people. So that's again a good event to organize or to attend here. So thank you, Ramesh, for your introduction. So let me start with this. So as already discussed by the principal, sir, that so we could see that biotechnology and its applications in human health. So mostly I would be focusing on the applications part, and so looking at the current pandemic. So I will take some examples of the specific pandemic related issues and all. So I will be talking about that. So before starting with the applications part, I'll be introducing you to basic biotechnology as already announced by the principal, sir. So I'll just briefly introduce you for that, and then I'll take you into the immunological aspects of our health, and then I'll talk about the applications part. So coming on to this today's topic, so we know that, as already emphasized by the principal, sir, so we could see that biotechnology is a combination of amalgamation of different subjects like technology, engineering, biology, and its applications for human welfare. So we could see that. So biology, so this pie chart explains you about biology, chemistry, and engineering technology. So biotechnology is at the pivotal point of all these three. So where we are using technology, chemistry, physics, and all subjects for the development of human applications. So it could be for development of agriculture, for development of human health, or whatever it might be. So in today's topic, I'm going to let's discuss about the different branches broadly classified, different broadly classified branches in biotechnology. So that's animal biotechnology, medical biotechnology, industrial biotechnology, environmental biotechnology, plant and agriculture biotechnology, and the last part is bioinformatics. So now the present, in the given present pandemics, we are more concerned about the medical and animal biotechnology and industrial biotechnology, where we are trying to create different diagnostic methods, our different therapeutics, our vaccines development and everything, and produce them at a industrial scale, so that can be supplied to the complete Indian population, as well as can be shared with other countries abroad. So for that, again, we need the different applications of bioinformatics, where we predict, or we try to predict, the exact combination which can be made artificially in a lab system, in vitro system, and then test that. So before testing that, so because of the short span of time and the urgency for vaccination and therapeutics in the present given pandemic, so we mostly depend on bioinformatics to predict a molecule or the structure of a molecule, whereby we can generate different therapeutics or vaccines against that, and then use it in a medical system. So there are different, I have just mentioned 6 different topics here, are branches of biotechnology here. So why I chose to give a lecture or deliver a lecture on medical biotechnology today is because of the importance of this one. So here you can see, so percentage wise applications are firms related to the different aspects of biotechnology, we can see here. So 51% of your share of your complete industry is taken away by medical and health biotechnology. So we are more concerned about our own health. So we are more worried about that. So we have 51% share of this one. Yeah. So that's the importance of medical biotechnology, where we are going to talk about today. So the others, you can see the agriculture biotechnology is around 12%. It comes next to the medical biotechnology. Then comes your industrial processing, like upstream and downstream processing, which counts for 9% of that. And others, so small other applications, is around 9%. And bioinformatics is gradually increasing. It's at 4% right now. So we might expect that to go up to almost 25 to 30%, so that it can provide leads and lead molecules and predict different molecules, and emphasize the or increase the rate of different applications in all other fields. So that's the special emphasis which is given to bioinformatics. So today I'm going to talk something about your immune system, how exactly it functions whenever you're infected, and then we'll talk about the applications part. So coming on to the immune system, before we go into the immune system, so we should know that, so we have hematopoietic stem cells which are residing in your bone marrows. So now these hematopoietic stem cells are responsible for
all the hematopoiesis. That means development of blood. So here we can see that the single hematopoietic stem cell that might give rise to two different lineages. That's myeloid progenitors, which give rise to all your blood cells like the dendritic cells, monocytes, neutrophils, eosinophils, erythrocytes and all. So which are a basic components of your innate immunity. And then we have lymphoid progenitors. So now which might give rise to your natural killer cells, the T cells and the B cells. So when you talk about the innate immunity and adaptive immunity, so the innate immunity is there are built in our body right from the time, from the moment of our birth. So that means by the time of your birth, you already have your monocytes, neutrophils, eosinophils, basophils, and platelets and erythrocytes, which are ready to encounter any antigen or a pathogen which is coming into your body, and they can fight that. So that fight is only naive, and it can only be restricted only to a particular level. But the adaptive immunity plays an important role. And this, as we already said, that it's an adaptive immunity. That means it has to identify that, and the system has to adapt to that and generate an immune response against that particular pathogen which is entered into your body. But this particular adaptive immunity has got memory. So it can be stored for future, where your complete system can be continuously monitored. And if the same parasite or a pathogen enters your body for the second time, so these adaptive immune cells will try to fight against that with a quicker response and a very high intensity of response there. So now when you talk about so we are more concerned about the adaptive immunity. So when you talk about therapeutics or anything, vaccines and all, so we are more concerned about the adaptive immunity. So let's focus on adaptive immunity. So the two different factions of adaptive immunity are the T cell progenitors, which give rise to the T helper cells and T cytotoxic cells, and the B cell progenitors, which give rise to antibody production in your body. So let's see how exactly the immune system functions. So just give you an overview of the immune system. Then I'll talk about the B cell development and the T cell development. And then we'll see how exactly we can manipulate these so that we can use our own system to fight against pathogens rather than introducing artificial chemicals into your body. So a successful immune response to a pathogen depends on the finely choreographed interaction among diverse type of cells. It's not like a single cell, a B cell or a T cell directly attacking a pathogen or an antigen and trying to neutralize that. So the first thing which happens when a pathogen or an antigen enters your body is the innate immunity is activated. That's what we talked about. So the neutrophils, macrophages, dendritic cells, these cells, they go bind to the antigen, they eat the antigen, they eat away the antigen, then they recognize that and they signal the adaptive immune cells to start an immune response against that. So if the intensity of your antigen or a pathogen is low, the innate immunity itself is more than enough to manage with that. But in case of, we already know that the pathogens like bacteria and viruses, they multiply at a very quicker rate when compared to our own cell division. It's like for every 20 minutes, we can find a bacteria to divide. So that means to counteract that, we need same level of soldiers to fight against these. So the innate immunity identifies the pathogen and shows that pathogen to the adaptive immune cells, that's the lymphocytes. So thereby the B and the T lymphocytes are activated. Once the B and T lymphocytes are activated, so the B lymphocytes start generating antibodies and the T lymphocytes start functioning in two ways. So one is the T helper cells, the other one is the T cytotoxic cells. So the T helper cells usually regulate the activity of all the B cells, T cytotoxic cells and all. And at the same time, they also have got a memory. So some of these cells can be stored for future for memory, whereby the memory lymphocytes, in case if you have any sort of infection again, so the memory lymphocytes take over that and they try to manage with that. So this particular point of memory lymphocytes is the basic rationale behind developing the vaccines. So that means we are triggering the B and T lymphocytes to identify a pathogen which is not infectious We are trying to kill the pathogen and inject that into the body so that the adaptive immune cells are activated and a memory is generated. So if the actual pathogen, which is infectious, if it enters your body, I do have memory cells which can easily identify that and fight against that. That's the basic principle of vaccinations. So let's see how exactly the lymphocytes are activated, are developed in your body, and then what are the applications, why, how we can use these. We'll talk about that. So coming on to the B cell development. So we know that I already said that hematopoietic stem cells, they are harbored in the bone marrows. So specifically in the larger bones like femur, humerus, or your uh, hip bone, or the sternum. So all these are the sites of hematopoiesis. So whenever you have these hematopoietic stem cells present in the bone marrow, so now depending on the signals they receive, they can transform into either myeloid or lymphoid origin cells, which might further give rise to different types of cells which are required. So whether it could be myeloid origin. So we know that already, we already know that your blood cells are replaced every 90 days. So that means every 3 months, you can see the blood cells are killed and new blood cells are generated. So that's completely based on the signal which the hematopoietic stem cells present in your bone marrow. They receive that, okay, this is the number of cells which are required and which type of cell is required. So the signal is communicated to the hematopoietic stem cells so that the hematopoietic stem cells, they start generating the specific type of cell which is required at that given point of time. So this is a continuous process with age. So gradually, if you have a healthy lifestyle, you don't have any problem with your hematopoiesis even at a late age. But if you don't follow a healthy lifestyle regime, so in that case, you might find that the hematopoietic stem cells are gradually replaced by adipocytes, and in old age, you might see an increased level of infection rate of infection, and you also can find some anemic conditions in later stages of your life. So that's because of your lifestyle, and the chemical signals which are transmitted or which are required for generation of different types of cells are either blocked or the quantity of the signal is lowered. So that leads to that. So here in this case, you can see that the hematopoietic stem cells, they are transformed into a lymphoid lineage cells, lymphoid progenitors. So now these lymphoid progenitors can either convert into a B cell or a T cell. So the B cell development completely happens within the bone marrow itself, and the mature B cells now leave the bone marrow, and they reach the different sinuses and lymphatic system and reach the peripheral blood vasculature in your body. So now they continuously move across your blood vessels and the lymphatic system so that they can screen for the different antigen presence or pathogen presence here. So if you talk about the B lymphocytes, there are millions of B lymphocytes in your body, and each B lymphocyte has got a specific receptor so that it can only bind a specific antigen or a pathogen. So these are randomly generated. So there might be a chance that some of these might even interact with your own body cells and your own body proteins. So to prevent autoimmune disorders, all these B cells which can interact with our own body proteins and cells are eliminated within the bone marrow, and only the mature B cells which doesn't interact with our own body cells or proteins will be sent out into the blood vasculature. So that's the process by which a B lymphocyte. So as I said that each B lymphocyte can bind only to a specific antigen or a pathogen. So here we can see that whenever, if you talk about any of our cells, like whether it's a T cell, B cell, whatever cell has. So each cell has got multiple different types of proteins which act as markers on their surface, present on themselves. So now all these proteins, if you inject them into a different organism, you find that all these proteins can act as antigens or epitopic sites, whereby our own B cell, if it is injected, let's say we inject that into a mice or a rabbit. So this will act as an antigen or a pathogen. So the mice immune system treats that as a pathogen and produces antibodies. The B cells in the mice, they start producing antibodies against our own B cells, and they might target different proteins, and each of these protein. So let's say if I have 10 proteins or markers, membrane markers present on my B cells, so each of them can be treated as a single antigen. So that means I have 10 different antibodies or 10 different B cells which can be activated against my own B cell in a mouse system. So that means this type of antibodies, a mixture of antibodies representing all the markers present on my B cells, we call them as polyclonal antibodies or polyclonal serum. So the polyclonal serum sometimes has got a very good effect, and sometimes it might be a problematic issue. So people, to cut short, I'll just say that during this activation, if I can identify the specific B cell which is producing a particular antigen, let's say we talked about the COVID virus, we know that there's a protein called as the spike protein, which helps in entry of that particular virus into your host system or host cells, infect the host cells. So it's receptor mediated interaction and receptor mediated internalization, which leads to the infection of your cells by the SARS-CoV-2 virus or the COVID virus. So now in this case, so if I want to identify to which protein the spike protein comes and binds so that it can internalize. So there is a protein called as ACE protein. So the spike protein comes and binds onto the ACE protein, which helps in internalization of this one. So if I can block this ACE protein or a spike protein, that might lead to a good therapeutic so that I can control the infection rate there. So for that, I need a specific antibody which can go and bind to the specific protein or identify and bind to a specific protein which is present. I don't need all the proteins which are present on that surface. I just need that protein to be blocked. So that means I don't want a polyclonal serum. Rather, I need a single type of antibody. We call it as monoclonal antibody. So now let's talk about monoclonal antibodies. So let's see. So I gave you an example where a cell has got 10 markers on its surface, and each marker can be used as a antigen, and antibodies can be generated against these. But in fact, if I just take only one protein, so there might be different epitopic sites on the protein itself, on the same single protein, and these epitopic sites can be, each of them can generate a particular antibody against them. So this type of serum, which contains multiple antibodies raised against a single molecule, pathogen or an antigen, we call it as polyclonal serum. So we talked about that. So let me show you how it appears. So here, let's say this is a complete protein complex. So there are different types of proteins which are different epitopic sites, like they're marked in green, red, blue, and all. So here you can see that. So when all these are epitopic sites. When you immunize this particular protein, that means you inject this particular protein into mouse model or any animal. So their immune system will start working against that. You can see an immune response where they start producing antibodies against each color-coded epitopic site which is present here. So these are the antibodies which are generated, and this serum we call it as polyclonal serum. So right now, let me say the blue color represents your spike protein of your SARS-CoV-2 virus. So in that case, I would be morely interested in having the blue colored antibodies to be produced so that I can use them for therapeutic applications. So I'm least bothered about the rest 3 antibodies which are being generated there. So in this case, I might trigger the production of this particular blue color antibodies, but as we know that human cells have got a life or a fate, so usually the B lymphocytes, they have a life of around 1 week to 15 days, not more than that. They don't live more than that. So how can I continuously generate the blue color antibodies? So to generate these blue color antibodies continuously so that we can harvest them and use them for different applications all throughout your life. So I need to manage to immortalize these blue colored cells so that they can continuously produce these blue colored antibodies. So this process of immortalization of your blue cell specific cells to produce antibodies continuously, we call it as generation of monoclonal antibodies, and that is done through hybridoma technology. So what is hybridoma? I already told you that. Here you can see. So I have a mice which is being injected with a antigen. So the mice starts generating B cells which can produce antibodies against a specific antigen or a specific protein which I am trying to inject into the mice. And now, as I said, that this particular cell type has got a lifespan of only 7 to 15 days, not more than that. So now, to make this immortalize this one, so I need to make a hybrid cell by fusing that with a myeloma or a cancerous cell type or a lymphoma cell. So what do? I try to fuse these two cells and make a cell, or we get a cell which is immortalized. At the same time, it produces the antibodies of the required type which I am looking for. So it's not easy. Why do two cells fuse together to say that, okay, I can produce this for you forever and I can live forever? So for that, there are basic, simple technologies of biotechnology which we use to hybridize these to make a hybridoma, so that we can continuously produce the antibodies in the culture-free media, and then we can extract them and use for different applications. So the basic rationale behind this particular monoclonal antibody or hybridoma generation is such that simple. So I have a myeloma cell. So myeloma, wherever you say oma, that's a cancerous cell, and I have a B cell which generates antibodies. So the myeloma cells, I choose a myeloma cell where the salvage pathway is knocked out. So we have two pathways by which the nucleotides are synthesized in your cells. So the one is the de novo pathway, where it assembles the small molecules, all the small molecules, and make the nucleotides there. And the salvage pathway is nothing but it uses intermediate molecules like hypoxanthine and other molecules, and they try to generate nucleotides like adenine, guanine, and all these nucleotides. So if a cell has to continuously divide, so I require more and more cell cycles to happen there. I need more and more cell cycles to happen. And if these cell cycles are happening, so I need more and more DNA to be synthesized. If I need more and more DNA to be synthesized for each and every cell division, so I need more and more nucleotides. So if these nucleotides are not synthesized, the cell automatically is killed. So here what we are trying to do is, so there is a gene called as the HGPRT. So this HGPRT is like hypoxanthine guanine phosphoribosal transferase. This enzyme is required for the generation of adenine and guanine during the nucleotide synthesis. So if this HGPRT gene is knocked out in your cells, in a myeloma cell, so it can only take the de novo pathway to generate nucleotides so that the cell can continuously grow for. Whereas the B cells are normal, they have both de novo pathway as well as the salvage pathway. So I can use these cells or try to fuse these cells by different methods. It could be electroporation based method, it could be pegylation, where polyethylene glycol can be used so that the cells can fuse with each other. So now I'm trying to compel the cells to depend on. So I need a selection media. I'm using a HAT media, hypoxanthine, aminopterin, thymidine media. So where the hypoxanthine and thymidine are supporters or promote the salvage pathway, whereas aminopterin blocks the de novo pathway. So when I fuse these cells, so I need to screen and select the cells there. So during the selection process, so if it's only the myeloma cell which is left out there, we can see that the salvage pathway is anyhow crippled in that, and the de novo pathway is blocked by the aminopterin here. So the myeloma cells can't survive because the nucleotide synthesis is not happening there. So the cell is non-viable, it will die. Whereas when we talk about the B cells, the de novo pathway is blocked, but they can still continue with the salvage pathway, whereby they can continue their life. But we already know that the B cells can survive only for 7 to 15 days if they don't fuse with the myeloma cells. So now the third type of cell which we have is a fusion product of the myeloma cell and the B cell, which can produce a hybrid cell. So even if you block the de novo pathway, the salvage pathway is activated by the hypoxanthine and thymidine, which are present in the selection media, and promote the cell growth of this particular cell type. So when you can see these viable cells, so now you can see that after a few generations, the B cells automatically are cleared out, and only the hybrid cells which can take the salvage pathway still survive in there. So I can select these hybrid cells now. And the first the foremost important point which we need to make sure is that these hybrid cells, during the process of hybridization, they might lose the antibody generation capabilities as well. So I need to screen for the antibody generation capabilities of your hybrid cells, so that these cells would be used or can be grown in culture media, so that I can use them forever to harvest the monoclonal antibodies. So if I harvest these monoclonal antibodies, so what is the application? As I already told you, there are multiple applications of these. Here you can see that. So most of your diagnostic methods for looking for different proteins, drug levels in serum and everything are based on monoclonal antibodies. If I just have an antibody against a particular protein, I can screen out and say how much quantity of protein is present in your blood serum during diagnosis, tissue typing and blood matching. So if we want to go for organ donations, I need to have a patient and donor tissue typing. Our MHCs and HLAs has to match with each other. Only then we can go for an organ transplantation. So for identification and infectious agents, if I have antibodies against a particular infectious agent or a pathogen, in that case, I can use that antibody to detect the presence of that particular infectious agent or any proteins related to the infectious agents in your serum to identify or diagnose that particular protein there. Then identifying a cluster of differentiation. So just you can give me any cell type in your body. I can just say what type of cell is that and what is that cell. So what are the specific things which we can do in that case? So that can be based on monoclonal antibodies. So it can be used in tumor identification of tumors. If there are some tumors, advanced tumors, where the cells migrate from the original site of tumor and go and reach a different site and start a secondary tumor there. So I can identify that to say whether it's the same tumor or is it something advanced tumor which has migrated from somewhere else. So identifying and quantifying the hormones, like these days we find most of us are deficit for your vitamin D presence in your body. So that hormones and vitamins, the purification and everything can be done, or identification can be done by using your monoclonal antibodies. So these are the different applications which we have for monoclonal antibodies. So we'll still talk about the different other applications. So let's talk about.
Hello,
Let's
Murali
talk about
Krishna garu, please mute yourself, sir. Murali Krishna garu. Murali Krishna garu.
Okay. So coming on to the second set of T cells. So we said that the B cells identify a particular. We can identify that the B cells can directly identify an antigen or a pathogen and then start producing antibodies against that. But for the B cell to do that, they require the help of the T lymphocytes. The T helper lymphocytes has to produce the chemokines and cytokines, which trigger the B cells. So though the B cell identifies the antigen, generate antibodies, still it gets a signal from the T cells. So that means the T cells play a crucial role in your adaptive immunity. So the lack of T cells will lead to immunocompromised state. So that's what happens even in case of your HIV infections. So during HIV infection, the T helper cells, so the AIDS virus, HIV, infects the T helper cells, whereby the number of T helper cells goes down and that leads to immunocompromised state. Otherwise, HIV is not a deadly disease. So it decreases your immune system so that thereafter, whatever you disease, you catch any disease, that would be extended and you can't get rid of that lack of immunity in your body. So now we need to understand how exactly the T cells function, and then how we can manipulate these T cells to target different diseases like it could be cancer, it could be HIV infection, or whatever it is. So let's see that. So now, as we discussed earlier, the B cells are the B lymphocytes. If anyone has got any questions, you can raise your hand and you can stop me at any point wherever you have a doubt. Specifically, students, if you have any questions or queries, you can always stop me and ask me if whatever you can't understand. Okay, so now the T cells, like we already discussed that the B cell maturation happens within the bone marrow, but that's not the same case with your T cells. So the T cell in a naive or immature state, they migrate to the thymus, and within the thymus, they undergo positive and negative selections. They develop the either into a CD4 positive helper cells or CD8 positive T cytotoxic cells, and then from the thymus, once they mature and everything, then they are sent out into the bloodstream. So if you talk about the number of T cells generated in your body, so the ones which you find, the millions of cells which you find in your body right now are constitute approximately only 5% of the total T cells which are generated in your body. Almost 95% of the T cells which are generated in your body are generated during the selection process, because they might have specificity to bind to your own antigens or your own proteins. If these T cells are left out into the bloodstream, in that case, your own cells will be targeted and you find many autoimmune disorders to be coming up in your body. So for that process, within the thymus, we have multiple rounds of selection, positive and negative selection, where all the self-reactive antibodies or self-reactive T cell receptor bearing cells are eliminated. That means approximately 95% of your T cells are eliminated, and then the rest of them are sent out. Even if any T cell skips this selection process, because it's in nature, you might have mistakes to be happening. It's so though these are error prone, not error prone, so there might be a chance that 1 out of a million cell might escape this particular process of positive and negative selection, which might start an immune response. That's why we find patients with autoimmune disorders, they have these either B cells or T cells which skip this selection process or elimination process, and they are sent out into the peripheral blood so that they interact with your own cells and start damaging them, leading to autoimmune disorder. So there are regulation mechanisms by which, so though if a positive or negative selection is skipped, the T cells can be regulated by a selection process. So this is a positive regulator for T cell activation. We can say here you can see a normal APC. So this is a normal APC, means antigen presenting cell. Like the B cells, the B cell directly can go and bind or identify an antigen or a pathogen directly on its own, whereas the T cells can't do that. They require the help of APCs. So the professional APCs in your body would be your dendritic cells, the macrophages, monocytes, or the phagocytic cells, which can engulf or eat away the pathogen or an antigen, digest that, show a part of that along with the MHC molecule, major histocompatibility molecule. Here you can see a red colored dot here in the center. So this is the antigenic peptide, and the blue colored cell is your T cell here. So now the T cell receptor can identify this particular peptide only when it is exposed along with the MHC molecule on the surface of APC. So that means antigen presenting cells has to show the T cell that this is an antigen and you are supposed to react to that. So that's the first signal. So this might happen even with your own antigens or your own cell products. So for that, the T cells have got a regulatory mechanism where they have another molecule called as the co-stimulatory interaction, where the APCs has got a receptor called as the CD8086 molecule present on its surface. That has to interact with, here you can see that there's a receptor present on the T cell, which is interacting with that particular molecule. So this we call it as the CD28 molecule. The CD28 molecules interacts with the CD8086 molecule present on the APC. So when these two interactions happen and the APC releases some cytokines or chemokines, these cytokines and chemokines are transferred onto the T cells. The T cells have got receptors for that. So now these T cell receptors bind to these cytokines and then increase the population of T cells, that specific T cell. So this we call it as positive regulation of your T cell activation. In lack of the co-stimulatory interaction of CD28 and 8086 or absence of this cytokine signaling, though the T cell binds or identifies the antigenic peptide, it can't be activated. It still stays in a non-responsive state forever. So that we call it as energy. So ANERGY. So the energy means the non-responsive state of your T cell, even in the presence of an antigenic peptide. If the co-stimulus and the cytokine signaling is absent, this T cell won't generate a response against that particular antigen. So your normal cells, though they express this particular antigenic peptide, though this can express their antigenic peptides here, because our regular cells in your body doesn't express the CD86 on their surface, and the co-stimulated cytokine signaling or the cytokine production is not happening here. So the T cells which have escaped the positive and negative selections in the thymus, which they are skipped that, though we have some people who have got this particular autoimmune disorders. Most of us don't show any autoimmune disorder because the co-stimulating interactions are absent in your normal bodies. So though there are some cells which are escaping our own immune selection process, but 90% of us or 99% of us doesn't show any autoimmune disorders only because of this positive regulator, absence of positive regulators. At the same time, similarly, there are negative regulators where the CD8086, rather than interacting with the CD28 present on the T cells, it can interact with the CTLA4, which is present on T cells. So the CTLA4 are the PDL and the PD1 and PDL-L1. So these are different negative regulators. So whenever there's a regulation of this one, or the CD8086 interacts with CTLA4, so that triggers the negative regulation of T cells so that the T cell can't be activated anymore. So why we could see this one is that in most of our cancers, the immune cells could not recognize the cancer cell because the cancer cells start producing the CTLA4 on their surface, which interacts with the CD28, 8086 molecules and all, so that the immune system can't be regulated or triggered to activate the T cells against our own cancer cells. Otherwise, the cancer cells has to be eliminated. So nowadays, people are trying to use this as a therapeutic to target cancer cells rather than giving harsh chemotherapeutic agents and all. So we'll talk about that application. So this work of CTLA4 and PD1 and PD1 ligand binding negatively regulates T cell activation. So this was awarded a Nobel Prize in 2008, I think 11, and that forms a basic therapeutic application in many regions. We'll talk about that in the next few slides. So any queries till this part? Anyone? Any queries till this part? So now coming on to the last part of my talk today. So the applications of biotechnology in human healthcare. So we can see what all applications. So the first one which I talked about is blockage of negative immune regulators for effective cancer therapeutics. That's one. Diagnosis of diseases. I'll talk about that. Recombinant protein production for different disease conditions. And the last one is tissue engineering and organ development for transplantations. So we already talked about these uses or applications of your monoclonal antibodies. We already talked about that. So coming on to this one, as I already told you, the CTLA4 binds to the CD8086 present on the APCs, so that the CTLA4 interaction with CD8086 triggers or stops the T cell to be activated. In most of the cancers, the T cells are interacting with CT8084 there. The CTLA4 is being used, and that particular antigenic peptide which is present on the cancer cells can't be detected because these cells are producing a lot of CTLA4 related regulatory molecules. So now presently what we are trying to do is that we are trying to block this CTLA4 receptor present on the T cells by using monoclonal antibodies. So I already said that the monoclonal antibodies can be generated against any protein. So I can generate monoclonal antibodies, and the CTLA4 is blocked, which is present on the T cells. So this blockage of CTLA4, what happens is the negative regulation or the negative interaction of CTLA4 with CD8086 will be blocked. Thereby, it activates the T cell to positively target the cancer cells and eliminate the cancer cells. So here we are not giving any sort of chemotherapeutic agent where you have many secondary complications like hair loss, weight loss, bone density loss, and different cell damages and everything, which is a regular activity in case of your chemotherapy. So we are not applying any of these chemotherapeutic agents, just we are using an antibody to block our own negative regulator of your T cells so that the T cells can target the cancer cells and eliminate the cancer cells. So similarly, the PD1 is another negative regulator. We can see here the PD1 is again being targeted, and the monoclonal antibodies, they target the PD1. Here you can see the PD1 is produced by the cancer cells are intelligent enough. They start producing more and more quantities of PD1 on their surface so that they can evade the immune responses as well as they can evade the apoptosis process or cell death process. So now here we are trying to target monoclonal antibodies against the PD1 molecule. So the monoclonal antibodies bind to the PD1 so that the PD1 is not available for interaction with the PD1 receptors. So the T cells automatically target the cancer cells because they are positively regulated and they can activate that and target the cancer cells. So this is what we call it as immunotherapy. We are using monoclonal antibodies against specific molecules and targeting them so that the other ways of using monoclonal antibodies therapeutics are immunotherapy is that. So we know cancer cells starts producing more and more quantities of specific proteins on the surface. If I can generate antibodies, identify the specific protein which is highly expressed on cancer cells, I can generate monoclonal antibodies against them. And these antibodies can be tagged with a toxin so that whenever this antibody interacts with that particular cancerous cell, it binds to the specific protein there. And then the toxin will be released at that particular location so that the rest of your body cells are not affected. Only the cancer cells can be affected and they can be killed. So whereby you can minimize the toxicity levels in case of your immune therapies. So these are called as targeted therapeutics, specifically targeting a specific tissue type or a cell type and target them and kill them. And then we talk about the diagnosis. Already the principal sir has mentioned regarding ELISA and its applications and all. So now in the present scenario, in the present pandemic condition, we are talking about more about RT-PCR test and all. What do you mean by RT-PCR? So the RT-PCR is nothing but it's a reverse transcription real-time quantitative PCR. So here we can see that we use the swab to extract the tissue from your nose and the oral cavities. So that tissue is dipped into a solution where they can isolate the RNA from that, and that RNA is extracted, and then we put a reverse transcription PCR and look for the amplification. If there is a presence of that particular gene related to a particular pathogen, then we can say that you have infection. So during these pandemic times, you must have noticed a lot of people saying that what is the CT value? So the CT value tells you about the cycle threshold. So the CT represents your cycle threshold, where the more quantity of RNA is present, the amplification of that product can be achieved with a minimal number of replication cycles or a PCR cycles. We can get that amplification. If we get just, so let's say I have only one copy of RNA in that. So it takes almost 30 reactions to get to a particular quantity or a threshold which can be detected by your system. At the same time, if I have 100 molecules of RNA, if you are infected and you have 100 molecules of RNA in your system, so that means I can achieve the same threshold with maybe with just 10 cycles. So the CT value at which you can detect this, prominently detect the presence of that RNA in your samples. So that's what is called as CT value. So in case of CT values around above 20 or 25, we treat them as very mild or very mild infection. Whereas if you have a CT values somewhere around 10 to 12, we say it is a intense infection and it's a active infection phase. So biotechnology is playing an important role even in the diagnosis methods, our diagnostic methods which we have here. So we talked about both therapeutic potential of biotechnological applications, and then we talked about the diagnostic potential of that biotechnological application. So the next example which I'm going to talk about is another therapeutic application. So we know that anemia is nothing but lack of RBCs or low number of RBCs or hemoglobin. So because of this lack of RBCs, what happens is the oxygen supply carrying capability of your blood goes down. So once the oxygen supply carrying capability of your blood goes down, your kidneys and others, they start secreting different hormones and others, which support or go back to the bone marrow to say that I need to generate more and more RBCs or erythrocytes. So this is a regular process which is happening. In some cases, in some patients, what happens is either the kidneys are affected or the bone marrow is affected in that case because of replacement, tissue replacements, or hip joint replacement, or all these replacements. So the hematopoietic activity might go down. So at the same time, the protein which is required for the hematopoietic stem cells to trigger the production of RBCs, we call it as erythropoietin. So that erythropoietin protein level goes down, whereby the level of generation of RBCs goes down. So in earlier days, what happens is, so they used to give them erythropoietin, which is isolated from some other animals like pigs or goats or some other animals. And that used to be a very costly injection or very costly treatment for them. So there are different types of anemias. So I'm just giving you one of the examples here. So now in this case, now with biotechnological application, what we could do is we have isolated this erythropoietin gene, cloned that into bacteria as the mammalian expression system, and we started producing erythropoietin artificially in the industrial setup. So now the cost of this particular erythropoietin injections came down drastically, so that's available even for the poorest in the country. So we can say now earlier the erythropoietin injection at the initial stages, it was costing somewhere around 10,000 to 15,000 per dose. So now the same injection came down to somewhere around 700 to 800 bucks for each injection. So maybe in future we might expect that even to come down or lower down even than this one, so that's even available at 100 rupees or 200 rupees per dosage can be seen there. So here in this case, by using biotechnological application in an industrial scale, we are trying to produce erythropoietin, which can be used as a therapeutic in case of anemic patients. So large quantities of erythropoietin is being produced, purified, and then packaged and given to patients on a regular basis. So this is again one more applications of producing therapeutic proteins by using biotechnological applications where we are concentrating. So that's why I said the medical industry, our health industry, constitutes around 51% of the total industry that's related to biotechnology. So then coming on to the last part of my listings. So now we know that because of different accidents or lack of tissues for transplantation. Many patients are dying on a regular basis because of lack of tissues for transplantations, organs and tissues for transplantations. Like it could be heart, it could be any other organ. So now the latest trend is that people are trying to take cells from the same patient and generate iPSCs, induced pluripotent stem cells, and then they put these induced pluripotent stem cells onto scaffolds, and these scaffolds can be used to generate tissues or organs, whereby that organs or tissues can be transplanted onto the required patients, so that we don't need to depend on any cadavers or donors for the organ or tissue transplantations. So the only thing is we need to control or create a proper environment, chemical and mechanical environment. So people are working on the biomaterials as well as on the cellular media and other structures where, so here in this image, you can see on the back of a mice, they could generate an ear pinna. So any patient who has lost a ear pinna during accident, so that can be generated in a mice model and can be easily transplanted. So we don't need to sacrifice or wait for a donor for this particular testing. So this is another applications of biotechnology in case of medical field. So with this, I stop here regarding my talk regarding the different applications of health and biotechnology. So if you have any queries, you can ask if you have any queries.
Okay.
Dear participants, if you have any doubts, you can ask him now. Any doubts?
Hi sir, it's Krishna.
Yeah.
Yeah, I am sir from 2006 to 2009 batch from S.V.K.P. Present as a senior microbiologist in Dr. Reddy Labs. Sir, from last 6 months, I have doubt from CT value. Yeah, a good explanation of CT value. My friend also get Corona CT value 3225 like that told me. But a little bit confusion. But nice explanation and very useful to information, sir. Thank you, Sri Krishna Jaya, sir.
Yeah, thank you.
Yeah.
Any other doubts or any other queries? Ramesh, can I transfer that onto host onto yourself, S.V.K.P? Ramesh, anyone? Any doubts?
No doubts. Dear participants, if you have any doubts, you can ask now, or you can type it in the chat box. You can ask your doubts without any hesitation. You can ask, and he can ready to clarify the doubts. Jaydev, can you explain about the autoimmunity?
So in case of autoimmunity, there are different types of autoimmune system, autoimmune disorders. We have two different types. One is systemic, the other one is tissue specific. In both these cases, what we find is that most of our selection processes which we have are supposed to be eliminating our own specific antigen specific or our own body antigen specific T and B lymphocytes. So they escape this process, they come into the peripheral bloodstream. So thereby they start an immune response against our own system. Like it could be T lymphocytes. So let me give you one example. So in some cases, your the pancreatic B lymphocyte, beta cells, that's your insulin producing cells. So they have the insulin producing cells and there are receptors present on these cells. So now your T cells identify these receptors and membrane associated cells on the pancreatic beta cells as antigens, and your T cells, the T cytotoxic cells are directed to go there and kill these. So in case of type 1 diabetes or autoimmune type 1 diabetes, what happens is these patients, the T lymphocytes proliferate into the pancreas, they bind to these pancreatic beta cells which produce the insulin, and they try to kill them. So that particular patient by birth is not diabetic, but still after a particular age, because of autoimmunity, all his pancreatic B cells or beta cells are killed. That leads to a autoimmune disorder, and that leads to a diabetic condition, type 1 diabetic condition. So where the production of insulin is completely eliminated. So in these patients, it could be a possibility that during the screening process in the thymus, the T lymphocyte, which can bind to the self antigen present on the beta cell of your pancreas, might have escaped the process of selection. It comes out, and so whenever this gets activated, when it comes in contact with your pancreatic cells, it gets activated, attracts other, it proliferates there and targets all these cells there. So thereby eliminating the complete elimination of beta cells, thereby leading to the autoimmune diabetic condition. So similar conditions are seen in different other diseases as well. So we could see, so in case of your what do you say, lupus, so there are antibodies which are generated against your own DNA molecules, RNA molecules, or proteins. So thereby, wherever you find these antibody immune complexes, it starts settling down in your bone, in your different locations, and starts different disease. So in case of systemic, the disease symptoms and everything might vary from one patient to the other one. Like in case of rheumatoid arthritis, rheumatoid arthritis. So that's the same condition. So you have antibodies generated against your own synovial fluids and everything which is present in your bone, in your joints. So that gets targeted, and because of loss of synovial fluid there, so it leads to abrasion of both the bones there, leading to damage of the bones there. So in each condition, it's a different this one. So this is systemic one. In case of arthritis and lupus, it's a systemic one. Whereas diabetes, thyroiditis, and there are other conditions which are more specific to the tissue or organ there.
Thank you, Jaydev. Thank you.
So I hope I could answer that. Yeah, any other questions? So I was expecting some questions from the students. Any other questions? Anyone? Don't worry, I'm not going to mark you. I'm not your teacher. So you can just treat me as your elder brother, because I am also from the same college.
So in absence of questions, we will move on to the next part, that is vote of thanks. So thank you for the elaborate explanation about the applications of biotechnology in an elaborate manner. Thank you, Jaydev. Now I request Dr. K. Lakshmikanthamma garu, head of the Department of Zoology, to present vote of thanks.
Good afternoon, ladies and gentlemen. On behalf of S.V.K.P and Dr.K.S. Arts and Science College Management, Principal, staff, and students, I propose a vote of thanks at the successful completion of the national webinar on biotechnology and human health, jointly organized by Departments of Biotechnology and Zoology in association with IQAC. At the outset, I express my sincere thanks to Dr. Y.V.V. Apparao garu, Principal of the college and Chairman of this webinar, for permitting us to organize this webinar and for his valuable opening remarks and blessings. I am very much thankful to Sri K. Sashikumar garu, Head of the Department of English and Coordinator IQAC, for his blessings and encouragement. I cordially thank Sri T. Ramesh garu, Department of Biotechnology and convener of this webinar, for his keen interest in arranging such a wonderful webinar. I make a special mention to express my sincere thanks to the resource person of today's webinar, Dr. Sri Krishna Jaidev Magani, Associate Professor, Department of Life Science, Shivanadar University, Greater Noida, Uttar Pradesh, for sparing his valuable time to deliver his inspiring and thought-provoking lecture. I'm thankful to Sri M. Sai Ram Garu for extending his technical support throughout the webinar. And finally, I express my deep sense of thanks to the college management, principal, staff, and students, and in particular, participants from various corners of the state for extending their valuable hearted support and monumental patience for the successful completion of the webinar. Thank you, one and all. పర్లేదండి అసలు ఫుల్ కోల్డ్ సార్ నాకు
Thank you, madam. Feedback link is posted on the chat box. You can fill in after filling the feedback, you can get your e-certificate. Sir, with this, we'll conclude this session. Thank you for patient hearing. Thank you, sir.
Thank you for inviting me, Ramesh.
Thank you, Jaydev.
Yeah. So most probably I will try to visit the place once if I find time.
Okay, Jaydev. It's a nice presentation.
Thank you.
You explained in a very elaborate manner in each and every topic. Whatever I have asked, that is about monoclonal antibodies and all these things, you have explained in a wonderful manner. And about cancer therapy, targeted cancer therapy, all these are very, you have explained. I have some doubts, you have clarified that doubts also.
Yeah. So is college working right now? The classes are going on, or is it a vacation period?
Up to 5th have vacation, and the college will reopen on 6th June.
6th June.
6th June.
Okay. So if I find time, I might come there. So let's see.
Okay.
I would like to
Sure.
come and interact with the students there.
Okay,
Sure
Thank
then.
you.
So, okay.
Thank you.
I can exit now. Yeah.
Okay.
Yeah.
Thank you once again.
Thank you, Ramesh. Okay.
Thank you. ఇందులో పెట్టలేదు పెట్టానన్నారు నాకు పంపించారు లింక్ నాకు పెట్టారా శ్యాంబాబు గారు
సార్ నమస్తే సార్ బాగుంది
థ్యాంక్ యూ
చాలా బాగా సార్ ఎక్సలెంట్
థ్యాంక్ యూ సార్ ఫీడ్బ్యాక్ లింక్ దాంట్లో చాట్ బాక్స్ లో పెట్టండి ఒకసారి మీరు ఫిల్ చేస్తే మీకు
సరే సార్
సర్టిఫికెట్
సరే సార్
ఆల్ ద పార్టిసిపెంట్స్ ఆర్ రిక్వెస్టెడ్ టు ఫిల్ ద ఫీడ్బ్యాక్ ఫామ్ దెన్ యూ విల్ గెట్ యువర్ ఈ సర్టిఫికెట్ వచ్చిందండి Thank you.